new strain of coronavirus originated, and this new strain was very lethal. And the patient who got infected by this coronavirus, a new strain in 2004, they had a lot of breathing problems. Okay, and that's why it was called severe acute respiratory syndrome, or SARS. Okay, SARS fortunately did not become pandemic. Why did not become pandemic? Because the SARS virus, coronavirus, was very lethal. Its lethality rate was very high, and it did not lead to asymptomatic patient. Okay, the current coronavirus, it leads to a lot of asymptomatic patient who can spread the disease and they do not know they are carrying the virus. But this 2004 coronavirus, which is also called SARS, severe acute respiratory syndrome, it was so lethal that about 20% of the people affected people died, and anybody else who was affected who did not die, they showed the symptom. And so the person could isolate themselves, and the disease did not become pandemic, it remained endemic. It stayed in China and spread to a few European countries, but did not become pandemic. Okay. Then another strain developed, and that strain was called MERS, Middle Eastern Respiratory Syndrome. It was discovered in 2012 in the Middle East. Okay. That also called respiratory problem. Okay. But fortunately, it also did not become a pandemic. It remained endemic even today, only in Middle East. Okay. And then came the current coronavirus. It developed or originated in 2019, okay, in the Wuhan province of China. Okay. And how did it originate is still a controversy. Most of the people think that it came from bats. Bats were carrying this virus. It was not causing severe symptoms in bats. But when it was passed on from bats to the human beings, it caused severe respiratory problems. Okay. And then it became endemic first, okay, and then it spread out of China or Wuhan into other parts of the world and became pandemic. Okay, why it became a pandemic? Because a number of people who get this virus, they are asymptomatic. They do not know that, but they are carrying the virus and they spread it. Okay, and its lethality it is not so high as that of earlier 2012 coronavirus, 2004 one. Okay? And therefore, all the people get affected, they do not die, okay, but they spread to a number of other persons, and that's why it became a pandemic. Okay. Now, in order to find the cure for the problem, stop the problem, it was important that we understand the structure and the life cycle of the virus. So the attempt was made to understand the structure of the virus. Okay. And as Dr. Govinji said, that you need to collaborate with the physicist and the chemist and the structural biologist. So to understand the structure of the coronavirus, the structural biologists, especially the protein chemists, they started working on the problem. Okay. And here's the structure of the coronavirus. Okay, you can see that. And this virus is a membrane-bound virus. All of you must know about bacteriophages. Okay, bacteriophages do not have a membrane, but most of the viruses that infect human beings, okay, who are pathogenic to humans, they have a membrane. And coronavirus is to human, and therefore it contains the membrane. You can see that it has a membrane. Outside the membrane, you can see that there are yellow structures, and this structure gives it a corona-like appearance. And these are the protein called spike protein. Okay. Now, this virus is a RNA virus, okay, and I'm going to tell you more about its structure in the next few slides. Okay, it's an RNA virus, and its RNA is positive stranded RNA. Now, what is positive stranded RNA? A positive stranded RNA is an RNA that can serve as a direct template for ribosomes. So it can act as an mRNA. There are a number of viruses that are negative stranded RNA. Those viruses, their RNA cannot serve as a direct template for protein synthesis by ribosomes. So it has to be first replicated to give the plus strand, and only then it can get translated into protein. But the strand RNA of coronavirus is a positive stranded RNA virus. And it is one of the largest viral genome in the viral kingdom. It's about 30 kilobase in size. And it codes for about 29 different proteins. Okay, what are the 29 different proteins that it codes for? They are of two types. Okay, so one is structural proteins, other are non-structural proteins. The structural proteins are the proteins that make up the structure of the virus, and the non-structural proteins are the proteins that help in the pathogenicity replication, and propagation of the virus. So I will tell you briefly about the first structural proteins. There are 4 structural proteins in coronavirus. Okay. The first I'm going to tell you about is the nucleocapsid protein. Okay. Now, what is nucleocapsid protein? As I told you before, the RNA of the coronavirus, you can see it here, and you can see it here. It is the largest RNA genome of any viruses. It's 30 kilobase in size. That 30 kilobase has to pack into small volume. Okay, to pack into small volume, it has to go compaction. It does compaction by forming a complex with the protein. Okay, and that protein that helps in compaction, it's called nucleocapsid protein. Now, our cells also contain a lot of DNA, and that DNA has to undergo compaction to fit into a small nuclear volume. And that compaction is by nucleosomes or through histone proteins. So in prokaryotes or in viruses, there are no histone proteins for compaction. So they have other proteins, and for coronavirus, this is a nucleocapsid protein. So second important protein of the coronavirus is spike protein. Spike protein is the most important protein of the virus from the infection point of view. Where it is, you can see that this is a virus. This protein protruding out from the membrane, this is the spike protein. And this spike protein gives it the appearance of a corona. Okay. And this is the protein by which the virus get attached to the receptor and the lung cells and get into the lung cell. So if the spike proteins can be inactivated, virus will not be able to enter the lung cells or any of the cell and will not lead to any infection. So all the efforts to stop the propagation of the virus or infect the virus, they have been targeting mostly the spike protein. I'm going to tell you more about that. Then the second protein is envelope protein. I'm going to tell you more about that. And then the membrane protein. So let's talk about spike protein more. As soon as the virus, it was discovered that coronavirus has become a pandemic, the effort was to find the structure of the 3D structure, three-dimensional structure of a spike protein. It turned out that a spike protein is not a single polypeptide. It has got 3 different polypeptides shown in yellow, light blue, and green color. Okay, and those 3 polypeptides are identical in that together, intertwine around each other, and they get attached to the membrane. Okay, now this protein is undergo post-translational modification. You might have heard of the protein undergo modification by phosphorylation, acetylation, methylation, a number of other different types of modification. Glycosylation is also a type of post-translational modification, where carbohydrate moieties are attached to the protein. You can see that this is the trimeric spike protein, and on the top of it, glycol moiety, carbohydrates are attached, dark blue. So this is the glycoprotein. And glycoproteins are very important. Okay, each cell of our body has a unique property. Okay, and that unique property is conferred by the glycosylation of the protein. So presence of a special carbohydrate on the cell surface make every cell very different from each other. And that glycol moiety or carbohydrate play a very important role in protein-protein interaction. And as you will appreciate soon, that spike protein has to undergo protein-protein interaction in order for infection to be completed. And this infection, I'm going to tell you soon on the next cycle, it is through the carbohydrate moieties. Envelope protein is this protein. You can see that it is present just below the envelope or the membrane. It is consist of 1, 2, 3, 4, 5 polypeptides. Okay, it helps in the maintaining the structure of the virus. The third protein, which is important, is membrane protein. And probably you have been taught by Rashmi or other people that membrane has got a lipid and the structural proteins. And those proteins are of 2 types, integral proteins or some protein that protrude out of the membrane. So this membrane protein of coronavirus, you can see that this is the 3D structure. Okay, it's an integral protein. Okay, it gives shape to the membrane of the virus. Okay, and that's why the virus shape is circular or spherical. Okay, now the nucleocapsid protein. If you remember histones, histones are positively charged. They contain the preponderance of arginine, lysine, and histidine. And because of the positive charge, they are able to bind to the negative charge DNA. Similarly, the nucleocapsid protein also has got the basic region. You can see that basic fingers and the basic palm. And through this basic region shown in blue here, it is able to attach to bind to the negative charge on the RNA to form the compaction of the RNA. So those charges, positive charges in the blue region, are important for the composition of the virus. Now, next talk about the non-structural proteins. Okay, non-structural protein, there are 15 proteins. Okay, and the function of all of them are not known. I'm going to tell you about only a few of them, which are very important. And the most important is the viral RNA polymerase. Viral RNA polymerase is activities in NSP12. It is responsible for the replication of the virus. So NSP12, this is the protein, okay? And this protein is associated with two other proteins, NSP7, NSP8. NSP stands for non-structural protein, okay? So NSP7, NSP8, and NSP12 together form the RNA polymerase complex. And this is a RNA-dependent RNA polymerase, unlike our RNA polymerase, which is DNA-dependent RNA polymerase. Now, this is the RNA-dependent RNA polymerase, which will bring about the replication of the virus. Okay. Now, there's another protein which is very important, and this protein is called NSP14. So NSP14 has a proofreading activity. Okay, our RNA DNA polymerase also has proofreading activity. That's why when our genome is replicated, DNA is replicated, there are very few errors because of proofreading activity. NSP14 also has proofreading activity. And this is unlike the flu virus, influenza virus, whose RNA polymerase has no proofreading activity. Therefore, flu virus, influenza virus can mutate very fast. But coronavirus, because the NSP14 activity does not mutate so fast, but still you hear that there are a lot of mutations. For example, the Omicron variant that was derived from Delta variant has got 52 mutations. And that was mind-boggling because of the proofreading activity having 52 mutation, that was difficult to digest. So a group in the Cambridge started working on the problem, and they found that the Omicron virus, it developed from Delta virus because of the recombination. So Delta virus was very lethal, okay, and it exchanged a big part of its RNA genome with the coronavirus that caused common cold. Okay, so it acquired all the mutation that the coronavirus, that common cold called, into it. Okay, and because of that, Omicron virus acquired all the characteristics of the common cold virus. And you can see that Omicron virus does not cause severe symptoms. Why? Because it cannot enter the lung. It cannot enter the lung. It is restricted to the upper respiratory tract. In the upper respiratory tract, it can cause sore throat, it can cause sneezing and cough, but not respiratory problem. Okay, so Omicron variant, in fact, was a blessing in disguise because it was more infectious. It was spreading very fast. But any person who was getting infected by Omicron was not having breathing problem, just having sometimes mild fever, sometimes only sore throat and sneeze, but the person used to get recovered, and person used to get the natural immunity. Very important thing. Okay. Now, in order to understand the life cycle, let's go to the how does the virus infect the cells. Okay. So this is a coronavirus, and it mainly infects the lung cells and the cells in the upper respiratory tract. In the lung cell, on the surface of the lung membrane, you can see that. Where is the you can see that this is a receptor. It's called ACE2 receptor, angiotensin converting enzyme 2 receptor, and this is the receptor where the coronavirus bind. Okay. How does it bind? The coronavirus bind to the receptor through the spike protein. Okay, I will show you in the next slide, and large view. You can see that. So this is the coronavirus, this is spike protein, and you can see that the dark blue part I told you, that's a carbohydrate moiety, and you can see that the principal interaction of the spike protein with the ACE2 receptor on the surface of the lung is through those carbohydrates. And once the complex is formed, okay, then the membrane of the virus fuses with the membrane of the lung, and the content of the virus, RNA and the RNA polymerase, are set inside the lung cell. Inside the lung cell, because this is the positive strand virus, it is replicated, it is translated immediately by the ribosomes to form the proteins. Okay, and the NSP12, which was there, okay, it starts replicating the virus. You can see it here. This is the ribosomes. Ribosomes are replicating the plus positive strand, and NSP12 along with the NSP768, it's also replicating the virus. So this is the plus strand and this is the negative strand. Both positive and negative strand are undergoing translation to form the proteins 29 different proteins. And those proteins get assembled to form the viral particle, which escapes the lung cells and they infect the other cells. Okay. Now, if you have to stop replication, you have to stop at this step or at the step of replication of the virus. Okay. Let's talk about various steps. Okay. So the two main points where the therapeutics have been designed to stop the replication or the propagation of the virus are blocking the entry of the virus into the lung cell or the cells of the upper respiratory tract or blocking the replication of the virus. Let's talk about one by one. Okay, so these are the monoclonal antibodies. Okay, monoclonal antibodies are very important. Okay, so those antibodies were developed against the spike protein. Okay, normally what happens? This is a SARS-CoV-2. It's bind to the AC2 receptor on the lung cell. Okay, and then the virus gets inside the cell. But if you have the antibodies, monoclonal antibody, then those monoclonal antibody can bind to the spike protein, and this virus, which is not totally surrounded by spike protein, cannot bind to the AC2 receptor on the lung cell. Okay, so you basically block the entry of the virus. Okay, and there are three types of monoclonal antibody. One from the Lilly company, other from Regeneron company, other from the GlaxoSmithKline company. So the antibody from Lilly and Regeneron, they were very effective against the Delta variant. Okay, but they were totally ineffective against Omicron variant. Okay, so then they developed the new antibody, monoclonal antibody, GlaxoSmithKline, okay, that's called sotrovimab, and those monoclonal antibodies were very effective in blocking the entry of the Omicron variant in the cell. Okay, but those monoclonal antibodies are very, very expensive. Okay, so there was a need for a simple, less expensive treatment, and that was in the form of the vaccine. Okay, vaccines are of three different types or four different types. Okay, the most important that all of you have heard of and which we are using in USA a lot are mRNA vaccine and viral DNA vaccine. RNA vaccine is what? RNA is that the RNA that is coding for spike protein. Okay, that RNA is packed into a nanoparticle of lipids. So this is called liposome, and this is injected into the cell, into the host, into the individual. It got injected to me also, probably to Dr. Govinji also. Okay, inside the cell, the liposomes are lysed and the RNA is released. The RNA is translated. You can see that this is the liposome. RNA is translated to make a spike protein, and when the spike protein produces the blood starts producing antibodies against spike protein. Okay, and those spike proteins then go and bind to the virus and prevent the entry of the virus into the cell. Okay, DNA vaccine similarly. Only thing, the DNA is packed into capsid protein, and DNA then is get transcribed into mRNA and then get translated. DNA vaccine is slightly better because mRNA get degraded, DNA can keep on making more mRNA, so there's a production of antibody for a longer time. The third category, so there are two types of vaccine, mRNA vaccine, Pfizer and Moderna. Both are very, very similar. The Moderna is slightly better because it can be stored at 288 degrees Celsius, but Pfizer can be stored only at very low temperature, minus 20 degrees Celsius. So the third category of vaccine is simply the protein, the spike protein itself. So recombinant protein was produced in moth cells, insect cells, and those purified protein were coated on the surface of a carbon nanoparticle. Okay, and those nanoparticles that are coated with spike protein, they were injected into the patients. Okay, so this is under clinical trials, and it has proved very effective. Okay, so because spike protein is going to produce antibodies, and those antibodies are going to bind to the virus and prevent the entry into the cells. Okay. This was about the entry. Now, there was another very important, promising drug called nafamostat. Nafamostat thought it's going to prevent the entry of the virus. Why? Because spike protein has to be trimmed or cleaved by this protease, TMPRSS22 on the lung surface. Okay. Nafamostat inactivate this protease. When it's inactivated, spike protein is not cleaved and cannot bind to the receptor. Okay, very promising, but it turned out to be that nafamostat does not work. Okay, there are many drugs which looks very promising, but they do not work because of some reason. Okay, but there's one drug which was very promising, which works, but with not such efficiency as was expected. It's called remdesivir. Okay, remdesivir does not block the entry of the virus into the cells. Okay, what does it do? It blocks the replication. Okay, what is remdesivir? Remdesivir is this small molecule. Okay, this is small molecule injected in the lung cells where it gets cleaved. You can see that two parts, it gets cleaved. And this yellow part is a nucleotide. It gets modified to form a nucleoside triphosphate. Okay, this nucleoside triphosphate is a nucleotide analog. It gets incorporated into the viral RNA, okay, during replication. But this is not the nucleotide. It's a nucleotide analog. So once this remdesivir get incorporated into the RNA replication stop at the elongation step, okay, and virus cannot replicate. In principle, looks very promising. It's supposed to stop the replication immediately, but it turned out that remdesivir is only 30% effective in replicating the virus. But when it's combined with other treatment, it's very, very effective. So what I told you about so far was monoclonal antibody, they prevent entry of the virus. Vaccine also prevent entry of the virus. Okay, remdesivir prevent replication. There's another nucleotide analog called melanoprevir. Japanese used it a lot. That also prevent replication. Now the last one, Paxlovid. Paxlovid is the treatment. Okay, what does Paxlovid do? Paxlovid is protease inhibitor. So when the viral proteins are translated, they are translated as long proteins, and they have to be cleaved to make the virus particles. And that cleavage is by proteases. Paxlovid, this reagent, this drug, can you see my arrow? This drug, it inhibits the proteases. Okay. And when the protein is inhibited, the long polypeptides are not cleaved, and the final protein product is not formed, which is required for assembly of the virus. So Paxlovid is also very effective, 90% effective, okay? But because it does not prevent replication, it prevents the assembly of the virus. Sometimes when you do the PCR test, you see the positive signals for that, okay? But it's very, very effective and very effective. Now, all those treatments are for one particular strain of viruses or one type of virus, coronavirus. Now, there's a need to find a cure that is not unique for one strain of coronavirus, all strains of coronavirus, but also other viruses also. So the emphasis is on finding something that can prevent the replication of not just coronavirus, but all other viruses also. Okay. Now, to find a cure, drug like that or cure like that, you need to understand how does the virus infect us. When the virus enters the host cell, our cell, human cells, what does it do? It regulates or controls the host transcription machinery. And this is transcription of the host cell. Transcription, you can see that this is RNA polymerase 2. Okay, RNA polymerase 2, it assembles on the promoter with the help of the general transcription factor. Then there's the elongation, and there's a termination. And then after that, RNA poly 2 is transferred to the promoter for reinitiation. Okay. Now, what does virus do? Virus hijack the host transcription machinery. Okay, virus transcription regulators, they make the RNA polymerase 2 transcribe their own gene, and they do not let the RNA polymerase 2 transcribe the host gene. Okay, how do they do that? They do that by targeting a number of transcription factors. Okay, it turned out that my lab started studying on that. Okay, and we found that a number of human pathogenic viruses, for example, herpes simplex virus, hepatitis virus, HIV, this coronavirus, SV40, cytomegalovirus, pox virus, all of them target one factor, and that factor is TF2B. TF2B. They target this factor, and when they are able to target this factor, they are able to completely hijack the host transcription machinery. They do not let this TF2B make the host protein, host mRNA, but they use it to transcribe their own mRNA. Okay, why they do that? Okay, so and it was so obvious that me and my student, we wrote a mini review article, the critical role of TF2B in viral pathogenesis. Okay, so my lab worked on TF2B. Okay, so we got a grant from National Institute of Health very recently to study why TF2B is the prime target of all the viruses. Okay, I'll tell you very briefly what we have found so far. So TF2B, probably Rashmi must have told you, or somebody else who's teaching you genetics has told you that TF2B is a general transcription factor. It's an initiation factor. Okay, so we started finding what is it doing that it becomes a prime target of the viruses. And we found that the TF2B is not just a initiation factor. It also plays a very important role in termination of transcription. After termination, it helps to couple termination to reinitiation. And once the polymerase is transferred to the polymerase for reinitiation, promoter for reinitiation, it makes the polymerase go in the sense direction to make mRNA. Because this polymerase can go in the upstream anti-sense direction, which is making a non-coding RNA, or it can do the sense direction making mRNA. So the TF2B not only help in initiation, but also in termination, but also in coupling termination to reinitiation. Okay, and then making the polymerase go in the right direction. So by targeting one factor, TF2B, the virus is able to completely control the host transcription system. And then it can make it transcribe its own protein, own mRNA, and then it prevents it from transcribing the host mRNA. And so if somehow TF2B can be targeted by some drug in such a way that it interferes with the replication of the virus, okay, then the problem is solved. It's not only one coronavirus or one strain, all the viruses that are shown here. All of them shown here, okay? They can be cured by this one simple tag. But the problem is that TF2B is also very important for the transfer of the host genes, okay? So the challenge is to find a drug that specifically inhibit the TF2B in such a way that it cannot be used by the viruses for transcribing the gene, but its ability to transcribe the host gene is remain unaffected. Okay, so that's the major challenge which we are trying to work on. Okay, I have a $1 million grant from NIH to work on this problem. We are collaborating with the pharmacologist. Okay, and whether we're able to do it or not, we're going to try our best. Okay, but it's a challenging problem, and we are on it. Okay, so far what I told you was done by my student. His name was Michael O'Brien, and my another student, Catherine Dwyer. Okay, so everything that I presented you from about my part of the work, which we are doing, that was done by those two students. Okay, and what did I do? I did all the talking. Thank you. Any questions?
That was indeed a very informative talk. Thank you, Professor Azhar, for igniting young minds by explaining about the science behind the COVID pandemic and its treatment and prevention in a very interesting and informative way. You have revealed the science behind the virus that we all have been impacted by. The platform is open for question and answer session.
Rashmi, are you asking a question? I cannot hear you. Yeah. If you can type in the chat, I can read it. I cannot, Rashmi, if you can increase your volume, then I will be able to hear you.
Is it better now?
It's better now. Or you can type in the chat. I can see the chat.
So I wanted to thank you for this very informative talk. And thank
You're welcome.
Young minds by explaining about the science behind the COVID pandemic and its treatment and prevention in a very interesting and informative way. You have revealed the science behind the virus that we all have been impacted by. I wanted to open the platform for question and answer session. So my students and my colleagues, they are typing the question in the chat box. If it is not, then maybe you can speak here.
Good morning, sir.
Good evening.
Any questions from any student?
Good evening, sir. Good evening, sir.
Good evening.
Thank you for presentation. And my question is, what is looking for the future of the pandemic, and when do you think it will come to?
If you can type in the chat, then I'll be able to answer your question. I cannot hear you.
I think there's some problem with the hearing from the other side.
Yeah. I can hear Govindji, but I cannot hear you.
Yeah. I had the same problem. I couldn't hear any questions.
Yeah. Okay. So I can see that now. What is the outlook for the future of the pandemic, and where do you think it will come to an end? Okay. So the outlook is good. Okay. It's be optimistic. Okay. There are a lot of cures, which was not there just 3 years back. Okay. Very good vaccines are there, and vaccines are getting more and more effective. Okay. And there's a lot of attempt to find a therapeutic. Vaccine are preventive, but you need a therapeutic. Okay. So Paxlovid is one of them, and there are other Paxlovids which are other therapeutic which are under clinical trials. Okay. So the future of the pandemic, this virus has now been reduced to merely a common cold coronavirus. Okay. It is not causing lethal symptoms in most of the individuals, healthy individuals, and those individuals who are dying or having severe symptoms, they are mostly immunologically compromised, or they have some underlying conditions like diabetes or some other things. Okay. Okay. But apart from that, that new strains, Omicron or other strains, they are not lethal like the earlier strain, like Delta and other. So, and one of the very important thing, anybody who's there who's not vaccinated, please get vaccinated. Okay. Even the Indian vaccines are very, very effective. Okay. They are traditional vaccines. Okay. But they are very effective. So anybody who is not vaccinated, please get vaccinated and get the booster if possible. Any other question? If you type it, I can see it very well. Yeah.
Have you put in more questions? They're putting in more questions to you. They're queuing for putting their questions on the chat. Yeah, okay, so now there are some questions. If you can see us. There are some questions in the chat box. If, can please read and answer the queries.
Yeah, Rashmi, if you can type it in the chat, I will see it very clearly.
Share in the chat.
Okay, so these days the people are affected with clots moving around either in the brain or affecting in the heart. Do you think it is the side effect of the medicine, or one who has been affected by COVID are having it? Okay, no, it is not the side effect of the medicine, it's the COVID effect, okay, of the virus. Okay, because when the virus is affected, the Delta strain, other strain, the immune system goes in overdrive. Okay, and that overdrive lead to the blood clotting. Okay, and then blood clot, it goes wherever it goes. It can go to heart, it can lead to the heart blockage. It can go to the liver, it can block the liver. Okay, so that can cause a lot of problem. Okay, so because of that, what they found a very simple thing, they start giving blood thinners. Okay, so the blood thinner, when they start giving blood thinners, that problem was completely solved. Okay, and that virus was causing the blood clotting, that was discovered very accidentally by Italian doctors. Okay, they found one person who died, and when they performed the autopsy, they found that he has clots that went to the heart, and the death was because of the heart attack or coronary thrombosis. Okay, so it was because the immune system going in an overdrive. Okay, and it was because of that. Okay? It was not a side effect of the medicine, it was the virus effect. Okay, and after they found that, they just start giving the blood thinner, and the problem was solved.
There are 2 more questions in the chat box.
How does the mutation in the virus can be studied and prevented? Okay, the mutation is studied by sequencing the virus. Okay, the virus is sequenced all the time in different countries. In India also it's sequenced all the time. Okay, and prevented. Mutation cannot be prevented. Okay, even though it has got a proofreading function, NSP12 14th subunit. Okay, but any DNA that is or RNA that is going to replicate a lot. With more replication, chances of mutations are always there. And the other thing is that it's not mutation, because there are a lot of coronaviruses which are floating around in our body. Okay, and their genome are very similar or homologous. So all of you know that in meiosis there is a genetic recombination. There is exchange of part of the chromosomes during meiosis. So similarly, there is exchange of the part of the coronaviruses, two different strains. And when the parts exchange, you acquire not one mutation, you acquire a lot of mutations, okay? And because of that, Omicron variant developed, it has 52 mutations over Delta, which was a parent strain. And those 52 mutations were not, did not make it more lethal. They made it more infectious, but the lethality decreases. Because of those mutations, it cannot bind, Omicron cannot bind to the ACE2 receptor in the lungs, and therefore you do not get the severe respiratory problem, okay? It remains in the upper respiratory tract and the throat, and it causes like a sore throat, cough, sneeze, some mild fever, nothing more than that, okay? And because of that, most of the people who are suffering from Omicron, okay, they recover very fast, and a very important thing, they developed natural immunity, okay, which probably is as good as the immunity acquired through vaccine. And if a person has got natural immunity and immunity through the vaccine, that person is the best protected. Next question, so given your knowledge in the topic, would you please tell whether it is possible that the virus can be artificially made and used as a bioweapon? Of course, virus can be artificially made, okay, and can be used as a bioweapon, okay. That is one of the concerns that's bothering everybody, okay, and it can be made. So there are regulations that prevent using viruses and bacteria as bioweapons, okay, but they can be used as bioweapons. You're right here. Can we make some vaccine to avoid any future COVID-19 similar outbreaks? Okay, so for that, we have to target something that is common to all the coronaviruses, okay? Because spike protein is most affected by mutations. Okay, if there's a drug or target that goes inside the cell and prevent the replication, okay, remdesivir is a very good one, but remdesivir on its own is only 30% effective. If remdesivir can be combined with proteases like Paxlovid, the two combination might be more effective. And that's why Dr. Anthony Fauci was talking about that a combination of 3 or 4 different drugs that might be very, very effective. Any other questions?
There's one more question coming in.
Okay, thank you. If you have any question, Rashmi, tell your students if they have any question, you have my email. You can ask them to email me, and I will respond within 24 hours. Yeah.
Can you hear me? Can you hear
I can
me?
hear you, Dr. Uni. Yeah, I can hear you very well. Yeah.
You can hear Govindji?
Yeah, I can hear you very well. Yeah.
Well, I want to thank you for your lecture and also for the way you were able to answer questions, which I failed. So it's very nice. Your system that you were familiar with worked very well. So thank you very much. I learned a lot from you because it's a totally
I
different.
learned a lot from
field for
you.
me.
Because, I've been
But,
learning from you since I was a student. I told you, I read your book, Rabinovich Govindji. I read all for, now I'm not working on photosynthesis, I'm working on RNA polymers all the time. But I still, when I get the chance, when I'm going through the journals, Nature or Science or something, if I find something on plants photosynthesis, I always read it. It's not that somebody's forcing me.
I learned a lot from you today, which I didn't know anything because I'm with plants. So it's very nice. Thank you very much for a wonderful talk and your decision how to handle the questions, which I didn't know how to do that. So thank you very much.
Welcome. Yeah. So Rashmi, what is the next step?
There's one more question coming in the chat box.
Okay.
There now.
Yeah, if you have any more question, I'm fully awake. Although it is how much the time? It's 1:30 here. I think Dr. Govindji is also awake.
Well, 12:30 for me.
Oh, you are in the second time zone. Yeah, I'm on the Eastern time zone.
12:30.
Yeah, I'm in 1:30. Yeah. So Rashmi, so if there are any other question, I'm very happy to answer any question from the students. Yeah.
Professor, I think there is a question in the chat box. Can answer that, please? You want me to read that out?
Yeah, you can write them chat. Yeah.
There in the chat box. There in the chat box.
Yeah.
And I also repeat that I'm very happy to answer questions, but send by email because it's easier.
Yeah.
So I didn't get any questions because I didn't know how to do it.
Not to worry. Thank you, Professor Atul, for sparing your valuable time to deliver this talk and answering all the questions patiently.
You're welcome. I welcome. And any student, anybody who has a question, please feel free to email me. Okay, Rashmi has got my email, and you can email me, and I will answer your question. Or if you need any other advice, okay, career advice, I want to do PhD. What should I do, and all that. Okay, you can email me, and I'll be more than willing to help you. Yeah. Dr. Govinji must have had so many Indian students to get the PhD and all that. Okay, and I'm in a position that I can also help. Okay, so if you have anything, feel free to contact me. Yeah.
Dr. Me and Dr. Roma and me, we are both especially happy to hear from you about polymerase, post-translational modification, and it instantly took us back to our Professor Satchar's lab.
You know the polymer that we're working with there in the Professor Satchar's lab. Okay, so that polymerase is in a complex with TF2B and a number of factors, and we were able to purify that whole complex. Okay, and that complex is doing transcription in a way that was not known before. Yeah.
Dr. Roma is now coming on the dialogue.
Who's it?
Roma here.
Roma?
How would you doing?
Roma? Roma Kapoor?
Yeah. No, but I need to talk to you one-to-one to understand more about it. But it is very interesting what you are doing. So, our students have written more questions in the chat box. If you are
Sure. Yeah, I will answer the question. Yeah, if you have more questions, I will answer them. Yeah.
they're already there in the chat box.
The chat. Okay. You said the immune system goes in overdrive and clots may happen. So those with compromised immune systems are safer than those with those with compromised immune system are less safe. Okay. And you said that the immune system goes in overdrive and clots may happen. So those with compromised immune system are safer than those with stronger immune system. No, people with a stronger immune system are more safe. Those with compromised immune system are not safe. Okay, and there's one more thing. So there are two types of immunities. Okay, T cell immunity and B cell immunity. Okay, so when you measure the B cell immunity, you measure the titer of the antibodies. Okay, and many people think that the antibody level is going down, so our immune response is decreasing. We are more vulnerable. That's not the case, because there's a T cell immunity, and it turned out that T cell immunity is more strong or more effective in countering coronavirus. Okay, and that is because of the compounds called lymphokines, which are secreted by T lymphocytes. Okay, and they are very, very effective. So the T cell immunity is also very important, and many of us have T cell immunity because we were infected by the common cold coronaviruses. Did I answer your question, Rashmi?
Yes, sir. Thank you. Thank you so much. With this, we conclude the third technical session of our two-day international seminar. I thank both the eminent personalities for sparing their valuable time to join us. Thank you so much. I thank all the participants for their patient listening and participation. Thank you very much.
You're welcome. Yeah. And thanks for the invitation, because it's an honor to talk to the students of your former university or alma mater, and especially Rashmi, you were there, you were invited. So again, thanks for invitation, and thanks for giving the opportunity.
Thank you.
I also want to say thank you, although I could not answer questions because I cannot hear what's being said there. So sorry about that. But please do remember to send me questions.
We surely will. Thank you, sir. Thank you so much.
Okay.
Totally blessed hearing from such eminent persons. Thank you very much.
Okay, you're welcome. Yeah. Thank you. Yeah. Thank you.
Is it okay if I end the session now?
Okay, so shall we leave, Rashmi?
Yes. Good night. Thank you for sparing your valuable hours. Maybe you move into the next day. Thank you. Okay.
Okay. Okay, thank you. Thanks. You're welcome. Yeah.
I can hear you very well, but not
No,
them.
yeah, I also can hear you, but I cannot hear Rashmi very well.
Yeah, there's something with the
Yeah, with the sound
system.
system. Yeah.
Yeah, right.
Okay, Rashmi, shall we leave now? Done?
Please, thank you. Good night.
Thank you. Okay, thank you. Okay. Thank you, Govinji. Thanks. Nice
Yeah,
meeting
thank
you.
you. Good
Yeah.
night to
I
you
also-
and
Yeah. Thanks a lot.
good
Again.
morning to them.
I met you personally in GNU long time back, so probably you not remember. I met you once, there was a photosynthesis workshop in GNU, long time, in 1980s. You came there, so we met you there also. Yeah. Thank you. Yeah. Okay. Thanks, everybody. Okay. And anybody, Rashmi, tell any of your students, if they have any question, they can email. You have my email. They can email and I will answer their questions. Okay.
And same for me, because very important for me, because I didn't answer any question. I didn't hear any question. So,
Thank you. Okay.
thank you.
Okay, Rashmi. Bye. Okay.
Bye-bye.
Bye. Yeah.